Hey, HL students. So we're going to finish off A1.2, which is DNA and RNA, by looking at some of the historical context of how DNA and RNA was discovered and what it meant for the knowledge that we currently have. So in the 1910s, initially, the structure of DNA was thought to be a tetranucleotide structure. And so there's an image of that here. There's a bunch of phosphates and they're all linked to adenine, thymine, guanine, and that they were having, they were always occurring in the same ratio. So there was an equal amount of guanine to cytosine as well as an equal amount of cytosine to thymine. But this was problematic because it didn't allow for DNA to have enough variation for it to convey genetic material. And so scientists at that time thought that the amino acids, the 20 different amino acids, were actually the genetic material. And so Chargaff did a study that was really quite important at that time, finding out the ratios of A's to T's to G's to C's. And what he found was adenine and thymine are going to occur in an almost one-to-one ratio in all organisms. And then guanine and cytosine also occur in an almost one-to-one ratio, but that thymine and adenine do not occur the same amount necessarily as guanine and cytosine. And so that was quite the breakthrough at the time, and it falsified the tetranucleotide hypothesis. Also at that time in the 1940s and 50s, which was a big time for discovering different information about DNA, McGarty and McLeod did this experiment with mice in which they showed that bacteria were able to pick up other bacterial genes somehow. At the time, they didn't know how. We now know that it's using plasmids. But the way that they showed this test was they took a non-virulent bacteria, meaning it couldn't kill the mouse, and they injected it into the mouse, and yay, the mouse lives. Then they took a smooth strain of the same bacteria, or of a bacteria, and that was virulent. And so they injected it into the mouse and the mouse died. Then they heat killed the smooth strain. So this bacteria is now dead, but it's got its genetic material in there. And they injected it into the mouse and the mouse lived because the bacteria was dead. But when they put the heat killed bacteria along with living bacteria, even though the living bacteria was non-virulent, what they found when they injected the mouse was that the mouse died. And so what the heck was going on there? It was that the bacteria that was alive was somehow picking up the genetic material of the virulent bacteria and using it and then killing the mouse. And then the other experiment we need to know about is the Hershey-Chase experiment. This comes up quite a lot on exams, so we should be familiar with it. And so this was to determine if DNA was in fact the genetic material. And so they were testing between protein or DNA or RNA. And so they took these bacteriophages and they radioactively labeled the sulfur in the proteins on the surface of one of the viruses. And then they radioactively labeled the phosphorus in the genetic material of the other virus. And so phosphorus doesn't occur in protein and sulfur does not occur in genetic material. And so they were testing to see if when the viruses infect the cell, if it's the protein that goes into the cell or if it's the genetic material that goes into the cell. And so if the genetic material is going to be DNA, then the bacteria will have that DNA, that radioactively labeled isotope in its structure. And so they ground up the cells after they were infected, and they centrifuged them, meaning they spun them around really quickly. And when they centrifuged them, the phosphorus, which is the heavier part that was in the bacteria, would have sunk to the bottom. And that sludge is going to be called sometimes the pellet. It is also sometimes called I'm totally blanking on the word. I will write it in the notes on the slide. But then the floaty part would have been it's called the supernatant. And that's the stuff that's not in the bottom, not the pellet. And so if the sulfur is in the viruses, that the sulfur in the viruses is going to be lighter. So you can see here, the viruses still have the sulfur on them. And that's going to be lighter, and that's going to be in the top of the tube, which is going to be the supernatant. So it's going to be floating in the supernatant. And then the bacteria, which are the heavier part, are going to have in the pellet, so sometimes called the sediment, is going to have the phosphorus. And that's what they found. Of course, because you're grinding up a whole bunch of stuff, you might end up with a little bit of phosphorus floating in the supernatant. You might end up with a little bit of the sulfur in the pellet, but realistically, you're going to have the majority of your phosphorus in the pellet, in the sediment, and you're going to have the majority of your sulfur in the supernatant, the floaty junk up above, because this is lighter. Okay, that's all for this video. Thank you for watching.